Kia ora koutou, ko subrusta takawingoa. Well, good evening, everyone, and a very warm welcome to you all to tonight's presentation on science and the healthier heart. My name is Sue Brewster, and I have the privilege of being the Executive Director of Auckland Medical Research Foundation. We're the largest independent funder of medical research in New Zealand across the spectrum of all diseases and conditions. I'm very excited to be introducing Professor Julian Payton and Dr. Nikki Earle, who will present their research in different areas of heart health tonight. First up will be Julian, an esteemed professor from the Department of Physiology at Auckland University and one of the founders of the Manaaki Manawa Centre for Heart Research. Julian's research into hypertension is truly cutting edge, and tonight you're going to learn how the nervous system is contributing to high blood pressure. You'll be shown how a heartbeat can be brought down from 154 to 54 beats, and how New Zealand is leading the world in the development of a pacemaker that is hoping to be able to reverse heart failure. Following Julian will be Dr. Nikki Earle, a research fellow in the University of Auckland Department of Medicine, and Nikki is focusing on the role that genetics play in heart disease. Nikki's study is looking at more personalised treatments with the aim of improving the stark statistic of 1 in 3 New Zealanders being readmitted to hospital, or even worse, dying after a second heart attack. So please sit back and make yourselves comfortable as I hand over to Professor Julian Payton for his 30-minute presentation, or thereabouts, on hypertension, earlier detection of stiffening of the heart, and a novel new pacemaker.
Well, first of all, I'd like to start by thanking the Auckland Medical Research Foundation for this fantastic opportunity to speak this evening. It gives me a great pleasure to talk about what we're doing in terms of heart research. The title of my presentation, At the Heart of Research, from Giraffe and Metronomes to 3D Printers, and that title will become obvious as we go through the talk. Now, I want to start off by telling you a little bit about cardiovascular disease in New Zealand. I think most of us know of somebody with cardiovascular disease and the consequences of that particular disease. In New Zealand, we have about 1 death every 90 minutes from cardiovascular disease, and that means 1 in 3 deaths in our country is caused by cardiovascular disease. Now, the mortality in our Maori population is at least twice that of our non-Maori population, and that presents an unacceptable inequity. And that's something we'd really like to resolve. Currently, there is no treatment for heart failure, which of course is a major goal and aspiration of what we're trying to do here at the University of Auckland. So how do we go about doing that? And to resolve that, we have set up a new heart research centre established in July 2019 called Manaaki Manawa. This is the Centre for Heart Research, located and hosted at the Faculty of Medical and Health Sciences at the University of Auckland. And I want to give a big shout out to our fantastic research operations manager, Lisa Wong, together with myself, who directs Manaaki Manawa. She's played a vital role for mobilizing this new heart research center. I'm just going to spend a little bit of time telling you about the center before I explain some of the exciting research that's going on. So the mission for Manaaki Manawa is, in partnership with Maori Pacific people, to create a world-class centre for heart research, underpinned by evidence-based and multidisciplinary research that delivers clinical benefits and equity to all in Aotearoa, New Zealand. One aspect of that mission statement is the multidisciplinarity, and that's quite unique because for the first time under our centre, we've been able to bring together researchers from different avenues. Classically, these researchers have always worked in silos. And so we've gone around breaking down the silos and throwing everyone into the same mix. So we have clinical scientists, biomedical scientists, epidemiologists, analytical scientists, bioengineering scientists, and biomarker scientists and genomics scientists, all working under the same roof, together with our Mari and Pacific cardiovascular researchers, so that we are fully integrated into a multidisciplinary approach to resolving some of the issues around cardiovascular disease inequity in New Zealand. This gives us an enormous strength. Manaaki Manawa actually stands for preserving the life force of the heart, and that name was gifted to us by Dame Nada Glavish. I'm going to tell you a little bit now about some of the exciting research that we're doing, and I'm going to block this into 3 compartments. I'm going to talk to you about a silent killer, then listening to nature, and growing your heart valve. These are 3 examples of some of the research that we're currently doing. I'm going to start with high blood pressure, which is a leading risk factor for death and disability in New Zealand. Now, around 20% of the population in New Zealand have high blood pressure, which is similar in other countries globally. As you may know, it's asymptomatic. It has no symptoms. Unfortunately, it's 32% higher in our Maori population. And remarkably, 50% of the hypertension in our communities can be prevented. It is indeed a modifiable risk factor. Let me talk a little bit now about risk factors. I think the first thing to say, and as you can see from this plot of blood pressure against age, is that blood pressure rises as you become older. And as blood pressure rises, it does indeed contribute to increasing the risk of a cardiovascular event, such as stroke, a heart attack, or heart failure, renal failure, and can worsen diabetes. Now, this risk obviously increases with age because blood pressure has increased, and high blood pressure is a risk factor for these comorbidities. However, if you also smoke or drink excessive alcohol, overweight, you lead a sedentary lifestyle, you have a high cholesterol or have high salt intake, and or have a family history, this further increases your risk of a cardiovascular event. That's the epidemiological evidence for cardiovascular events. And it's really now a matter of trying to understand what we can do about this. First of all, I want to tell you something that's come from some very recent data published this year about a disease called heart failure with preserved ejection fraction, which is a disease that's caused by high blood pressure. And as you can see in the top left-hand panel, we have a schematic of the heart. And what high blood pressure can do to the heart is thicken the walls of the main chambers of the heart. These are the so-called ventricles. Not only do they become thicker, but they also become stiffer, which means when the heart tries to fill properly, it can't fully dilate to allow new blood to come into the heart before it then contracts. Now, it may well be that under resting conditions, where you're not exerting yourself, you're okay, but it's when you start to exercise, when you need your heart to start pumping more blood, that the heart really struggles because it cannot fill to accept more blood, to pump more blood, because it's become stiff as a result of the high blood pressure. Now, we know this stiffening is caused by something called fibrosis, and it's an increase in fibrosis. And you have underneath the heart here, you have pictures of the heart at very high magnification, showing in red, the muscle cells, and in blue, the fibrosis. And you can see in somebody with heart failure with preserved ejection fraction, there is a lot more blue, a lot more fibrosis. Now, I'll tell you in a moment what that fibrosis is. But first of all, I just want to let you know that if we look at a single heart muscle cell as depicted here at the top right, every heart cell has little tubules that project into the inside of the heart cell. And this is important to allow the heart cells to contract properly. What we've detected, and this is work from David Crossman's group, is that in these tubules, there is a lot of fibrosis, and that fibrosis is due to something called collagen 6. Collagen 6 is a peptide. It's a important protein that helps hold things together. In the healthy condition, you can see here in green the tubules running in to the heart cell. You can see there's a little bit of red, and that's the collagen 6. But in heart failure with preserved ejection fraction, you can see that the amount of collagen here is greatly enhanced, which is causing it to become stiff. Now, the new finding is that we've detected a new biomarker for heart failure with preserved ejection fraction. And this is really exciting and important, because if we can catch it early, we can do something about it before it's too late. Now, during the formation of collagen 6, it releases a protein into the blood called endotrophin. And we can now detect endotrophin in the blood. And this recent study shows very nicely that if we look at survival of patients with heart failure with preserved ejection fraction against time over about 4 or 5 years, you can see those with high amount of endotrophin don't survive too well. Those with lower endotrophin survive much, much better. So the early detection of heart failure with preserved ejection fraction is now possible by measuring endotrophin in the blood. This will help, indicated beneath here, preventing worsening of this condition. But clearly, further research is needed to assess what we can do to actually lower collagen 6 in the first place and prevent stiffening of the heart. Okay, I'm going to move on, and I'm going to ask a question now. What was the greatest invention to lower blood pressure? Surprisingly, it was refrigeration. And the reason it was refrigeration is because the development of cooling food prevented its preservation by addition of salt. This tells you something about a major cause of high blood pressure, which is high salt. The other thing that helps to reduce high blood pressure is exercise. And I just showed this 24-hour fitness center here in the United States with a bit of a tongue-in-cheek, because you will notice that there is an escalator up to the fitness center, both up and indeed back down again too. I think only in America would you have a fitness center with escalators. Now, regarding the salt and high blood pressure, most of our drugs that are given to control high blood pressure are acting on the body to try and help remove salt and reduce the amount of blood volume by excreting water from our body. So if we look at treatment, which is typically A, B, C, D, standing for ACE inhibitors or angiotensin receptor blockers, beta blockers, calcium channel blockers, and diuretics, you can see how these drugs relate to different parts of the body. And the main organs that are affected are the kidneys, as you can see, the arteries, the adrenal glands, and the heart. A, B, and D will all help reduce the salt and water in the body, and they will counteract a hormone called angiotensin 2, which I want to come back to in a moment. Now, angiotensin 2 is a hormone. It's been in animals for many, many years. It was originally evolved when animals moved from an aquatic environment out into land, because when you move from water dwelling to land dwelling, you need to preserve water and salt because you don't want to lose water through dehydration. However, angiotensin 2 is a major culprit for cardiovascular disease. Unfortunately, both in New Zealand and many other countries around the world, blood pressure is poorly controlled. And there are a number of reasons for this. First of all, we notice that 50% of those that are treated for their blood pressure, so they're taking their drugs, still remain with hypertension. Although blood pressure may be lowered, they remain hypertensive. Those that are treated and controlled, that have normal blood pressure, remain at elevated risk for disease. And that is quite an alarming fact. This would suggest that our current drug armory is not preventing causes of high blood pressure, but rather treating symptoms. So the question then becomes, So what are possible causes for the high blood pressure? And if we were able to treat those causes, we ought to be able to have a most or more effective way of lowering blood pressure in patients. So I want to return to the idea that the nervous system, the so-called autonomic or automatic nervous system within our body, is contributing to high blood pressure. Indeed, the heart, as you can see here, is covered in nerves. The nerves are in red, and this is a picture of the heart. This doesn't matter if it's a human heart or an animal heart, there are nerves. Now, these nerves are part of the parasympathetic nervous system because there are two types of nerves that innervate the cardiovascular system, those that are parasympathetic and those that are sympathetic. And we see clearly here a very, very rich innervation by the parasympathetic nerves carried in the vagal nerve that goes to the heart and originates from the brain. Now, I'd like to demonstrate with a short video the connection between the brain and the heart. And the way I'm going to do this is I'm going to activate the vagus nerve to the heart, and the vagus nerve causes a slowing of the heart. And the way that we can activate that vagus nerve is to stimulate cold receptors around your face, around your nose and your lips. Now, these cold sensors are connected to a different part of the brain, which eventually connects to the vagus nerve and activates it to slow the heart. And this is called the diving response. And so when you put your face into water, your heart rate will slow down. And the reason it slows down is to preserve oxygen usage, because the body realizes that you can't breathe underwater. So this is a video of a participant that's going to put their head into a bucket of cold water while we record heart rate using an oximeter that's connected to the index finger of her right hand. And what I want you to do is I want you to listen to the beeps that this machine makes, which indicate her heart rate. Continue listening despite the fact that she will lift her head out of the water after about 20 seconds because her heart rate continues to fall. As you will have seen, her heart rate fell from a heart rate of 115 down to 54. That's a reduction of 61 beats per minute, and that is due by activation of this vagus nerve. Now, the other nerves I want to talk about that connect the brain to the cardiovascular system are the sympathetic nerves. And here, what we've got is a blood vessel. Every blood vessel in the body is connected to the brain via these sympathetic nerves, and they're these green wiggly lines. What do those nerves do? Well, those nerves, if activated, as you can see in this schematic, cause your blood vessels to constrict, become narrower. And as you constrict those blood vessels, that increases blood pressure. The analogy I would give is like putting your finger over the end of a hose pipe. You do that and you increase the pressure in that hose pipe, which means now you can squirt the water further from the end of the hose. You also know that the pressure is increased in the hose pipe because if your hose pipe is like mine, the other end of the hose connected to the tap normally blows off. So if we could find a way of reducing that activity in those sympathetic nerves, that should dilate vessels and lower blood pressure. What we've discovered is an organ called the carotid body, and the carotid body drives up sympathetic activity in those nerves to cause hypertension. And we demonstrated this for the very first time some years ago by selecting patients with drug-resistant hypertension. That means they were not responding to drug treatment and had huge pressures of around 180 to 200 millimeters of mercury. We removed one carotid body for the very first time. Here it is taken out. And reduce blood pressure in those patients by around 20 millimeters of mercury, which is quite a substantial lowering. The exciting news that I bring today is that we have unearthed a natural way to temper the activity of this carotid body. That means we do not advocate going around and removing carotid bodies. So we sought after to try and find a natural occurring compound to block those receptors, and have found one, but I can't tell you exactly what it is because it's under patent. The giraffe has also been a fascination of ours for quite some time because it has high blood pressure, but this is natural for it. It has blood pressure that's twice that of humans. Typically at the level of the heart, our pressure would be around 95 millimeters of mercury. But it needs that high pressure in order to push blood up its neck into its brain. And we've wondered for many years what it is about the giraffe that allows it to be able to adapt to high blood pressure, because it never dies of cardiovascular disease. Normally, unfortunately, giraffes die from being caught by lions. So what is it that we can learn? Well, the first thing we know is that the neck artery is wide and open, as you would expect, but those arteries in its leg are very, very narrow, and this is how it generates its high blood pressure, through a narrowing of the arteries in the leg. Those that are supplying the brain, such as the neck arteries, are nice and wide and open. And in fact, if you remember, if you increase the resistance to flow, you will get higher blood pressure, as we saw in the hosepipe. Now, very recently, the genome of the giraffe has been fully sequenced. And what's come out of that is something called fibroblast growth factor receptor like 1, or FGFRL1. This is a novel gene that's been identified and is related to high blood pressure. What's recently been published in Science is that that gene has a number of mutations, and those mutations relative to the human FGFRL1 are of unknown function. So in order to determine what the function of those genes those mutations in that gene might be, the giraffe FGFRL1 gene has been snipped out of the giraffe genome and has been replaced in the mouse genome, as indicated by the schematic. So now we are going to look at what happens in a mouse that now has the giraffe fibroblast growth factor receptor like 1. Here you see the mouse in the lower panel here relative to a mouse containing its own FGFRL1. Interestingly enough, you don't see a long neck. In fact, you don't see too much difference. If anything, the mouse is a little bit shorter at the same age. But what we do know is that the bone density is massively increased, which you would expect in a giraffe that's bearing all that weight. But there's something very important that's been discovered, and that is this. Remember I told you that angiotensin II is a major culprit for raising blood pressure. And indeed, if you look at the mouse with its own FGFRL1 and you give that mouse angiotensin II, you can see you can raise its blood pressure relative to giving vehicle control. If you then take the mouse that has the FGFRL1, surprisingly, angiotensin II is completely ineffective. That was something that had not been predicted. But what that tells us is that the giraffe is desensitized to angiotensin II, probably to protect itself from generating further high blood pressure. This opens up huge avenues for us now to exploit and to try and understand how we might be able to modulate FGFRL1 in humans. The next thing I want to briefly mention about blood pressure is renal denervation, and this is a one-time procedure to lower blood pressure that may help to combat issues around accessing clinical treatment for blood pressure, may also address compliance of drug taking. People don't like taking pills, and also tolerance issues around drugs that gives nasty side effects. What you can see on the left is the catheter that's placed in to the renal artery. It's an ablation catheter. It's a 2 or 3 hour procedure, and it ablates all the nerves going to the kidney. Now, when you do that, as you can see here, very recent data, after 3 years, you can see quite a nice fall in blood pressure, approaching 20 millimeters of mercury. We are currently hoping to set up a trial in New Zealand very shortly to be able to perform renal denervation. I want to move now to listening to nature, which is a new pacemaker that we're developing. Most pacemakers are situated as a device just under the skin on the chest and have a lead that goes down into the heart, as you can see here. That's very standard. The other thing that's very standard is that the pacing is typically metronomic. Now, the heart never beats metronomically. And so we raised the question very early on, why are we pacing hearts metronomically? And I want to demonstrate that hearts don't beat metronomically by looking at this participant and the way he breathes and how his breathing is affecting his heart rate. What he's going to do, he's connected up to the same device, so you will hear beeping again. Every beep is a heart rate. And I want you to listen to how his heart rate increases as he raises his hand. That's because he will inhale. He's indicating breathing in through raising his hand. And then as he breathes out, he will lower his hand and he's going to take 3 breaths. And those 3 breaths are going to be related to changes in heart rate, as you will hear. So we have developed a pacemaker that allows us to modulate heart rate every breath. And we found some spectacular results in a large animal model of heart failure. This is a sheep model of heart failure. We fitted our novel pacemaker such that every time the animal breathes, its heart rate increases, and every time it breathes out, its heart rate decreases, just as we saw in the video. And if we do that for a number of weeks, 4 weeks in total, you can see how the pumping, which is rather reduced because of the heart failure, dramatically increases by about 25%. That increase in heart pumping efficiency is 3 times that of current pacemakers, which really is a very exciting and novel observation. The question is how is it doing that? What we've seen when we look at the heart tissue, and these are pictures of the heart tissue that have been labeled with antibodies connected to fluorescent markers. One is red, indicating the T tubules. Those are those tubes that I was talking about early on. They're in red. And associated them with is something called ryanodine receptors in green. And now these ryanodine receptors are really important for contractility of heart muscle. They give it the power to contract. You can see in healthy situation, the green and the red are beautifully aligned, tubules with the ryanodine receptors. But in heart failure, the tubules become fragmented and the ryanodine receptors lose their location with those T tubules. But after our pacing, you can see this beautiful realignment of the T tubules and the ryanodine receptors, which means to us that we've actually beginning to see a way of reversing heart failure, reversing the damage to heart muscle cells. I'm delighted to say that we're able to start a trial in the first quarter of next year, which will be performed by Dr. Martin Stiles in Waikato Hospital. We will be using this new pacemaker connected to an external pacemaker device, which they use in the hospitals already, which will connect it to patients after they have had coronary artery bypass grafting. The reason for selecting those patients is that they have exteriorized pacemaker leads, which allows us to connect our pacemaker device directly to the patient without having to implant it. Hopefully, this time next year, we'll have some positive data on this new form of pacing. But what this does mean is that New Zealand has an opportunity here, if this works, to demonstrate to the world a completely new revolution of cardiac pacemakers. Now, I want to end with rheumatic heart disease. And rheumatic heart disease is something that this country, as a developed country, really should not have. You may know that rheumatic heart disease is due to repeated streptococcal or sore throat infections. After replication of these infections, an autoimmune disease is triggered, which means that one's own immune system now begins to attack the body. The part of the body that it attacks, unfortunately, are the valves within the heart. Now, a valve in the heart plays an essential role because it allows the correct direction of blood to flow through the heart. Rheumatic heart disease basically destroys these valves, which no longer operate, and therefore there is poor directional movement of blood. It doesn't know whether to come forward or back. And as a result, the heart becomes a really very weakened pump. Currently, to address this, patients undergo operations to either fit a titanium valve, a metal valve, or a valve from an animal such as a pig or cow. This is a real-world problem. Matt Johnson, who's a retired Blues midfielder, was diagnosed with rheumatic heart disease not so long ago. He no longer is able to play rugby because he has now been fitted with a titanium valve. And indeed, I met Matt not so long ago after his operation, and you could hear this valve click, opening and closed with every heartbeat. The other thing that Matt unfortunately has to do is take blood thinning drug called warfarin, because with a valve such as this, it can cause blood to clot. Being on a blood thinner means he cannot play contact sport because he's very prone to bruising and large bleeds under the skin. So this is hugely problematic for people clearly throughout their life. Now, rheumatic heart disease, if caught early, typically can be found in young children. And here we have a young man who has just had his rheumatic heart disease valve repaired. And this is 6 weeks since surgery. Unfortunately, for this young man, he's going to have a lifetime of operations to replace his heart valve. And the reason for this is that there are a number of problems. The first is the current valves don't grow. I showed you a metal valve and a porcine valve. They simply don't grow, but his heart will grow. And as his heart grows, so the valve will begin to leak because it will remain the same size. Very often, animal valves are rejected eventually by the body or simply wear out after a period of time. And so repeated heart surgeries are needed. And every time this child comes back in for a heart surgery, the surgeons are faced with technically more challenging operations. So what can we do to address this? And I think this is a nice example now of how Manaki Manawa, with its interdisciplinary approach to cardiovascular disease, really demonstrates its power. So recently, colleagues in engineering, Olaf Diegel, have received a 3D printer which allows us to print growth matrices and scaffolds that will support human tissue growth. This will be ideal for making personalized body parts. And as I said, interfaces engineering with medicine very beautifully. So here's what we're planning on doing. We're trying to grow your own heart valve. Let's imagine we have a young child that has been diagnosed here with rheumatic heart disease. We take a skin biopsy from that child and we reprogram the cells to produce from their skin cells pluripotent stem cells. We can then take those stem cells and drive them into heart valve cells. Now, at the same time, the child's heart can be scanned using high-powered magnetic resonance imaging so that we can get an exact copy of the size and shape of the heart valve needed for that particular individual, so-called personalized medicine. Once we've got an image, a 3D image of the valve, we can then set up the 3D printer to print an exact replica of a heart valve printed in this growth matrix gel scaffold. And now that gel will be impregnated with all the right growth factors that are needed to stimulate the stem cells to grow into heart valve cells. Those heart valves will then be implanted back into the heart of that recipient. And the beautiful thing here is that those heart valves will not only fit perfectly because they've been personalized, but because they have been grown from cells that originated from the patient, there will be no rejection of those valves. That is an aspiration. That is something that we would love to be able to progress to do with funding that could come from the Auckland Medical Research Foundation. So thank you very much for listening this evening. It's been a great pleasure to be able to update you with some of the fantastic research that's going on in Manaaki Manawa. Thank you.
Wow. I'm pretty sure that all of you, just like me, would have gleaned so much learning from Julian's presentation and from the research that him and his team are undertaking. It's pretty incredible stuff. Actually, one handy learning that I take away from all of this is that if I'm a bit nervous and my heart rate's going up, I could just stick my head into a cold bucket of water. But look, in all seriousness, incredible work that they're undertaking here. And look, Julian will be joining us after Dr. Nikki Earle's presentation. So where both of them are going to be online, and you just have to type your questions into the chat function, and they'll do their very best to answer as many as they possibly can. So stay with us as I welcome Dr. Nikki Earle, who's going to help you understand the risk factors of heart disease and how our genes influence our likelihood of a heart attack.
Hi everyone, I'm Nikki Earle. I'm a research fellow at the University of Auckland. Thanks very much for joining us this evening, and thank you very much also to the AMRF for this opportunity to talk about heart health and some of our research, and I look forward to answering any questions you might have at the end of my talk. So just to briefly introduce myself, I work in the Heart Health Research Group at the University of Auckland, which is led by the Heart Foundation Professor Rob Doughty. I originally studied genetics down in Otago, and then I tried a few different things and travelled overseas. And then I found my way into heart research, and I did my PhD on genetic causes of sudden cardiac death. And I was supported by an AMRF doctoral scholarship over that time. And I now work as a research fellow and am again sorted by the AMRF with a Douglas Goodfellow postdoctoral fellowship. So we're a clinical research group. So we undertake studies where we're enrolling people with different types of heart disease and collecting a lot of information from them. And this is often in the hospital setting after an event such as a heart attack or an admission for heart failure. And we then collaborate with lots of other scientists and research groups to measure biomarkers on blood samples that we collect. And we also do different types of cardiac imaging on our study participants. So a quick outline of what I'm going to talk about this evening. I'll introduce some of our research and then talk about how this relates to the genetic risk of heart disease and to women's heart health, which are both areas of interest for me. And then I'll finish off with where we're headed next. So heart disease can affect people across a lifetime, and that's beginning right from babies that may be born with structural heart problems. And our research is also a program of work across different parts of the life course, but I'm just going to talk about part of it tonight. And heart health is a topic that should be important for everyone. There are currently 175,000 people living in New Zealand with heart disease. So almost everyone has someone in their friends and whānau who are affected. 1 in 3 deaths in New Zealand are caused by cardiovascular disease, and that means that someone dies from it every 90 minutes. And many of these deaths are premature and preventable. Also, unfortunately, there are persistent inequities by ethnic group and level of socioeconomic deprivation, and Māori and Pacific peoples are disproportionately affected, and they often develop disease at younger ages. So the research I'm talking about this evening is focused on coronary artery disease, which is when plaque can build up in a coronary artery that supplies blood to your heart muscle. And this can eventually lead to a heart attack where the blood supply to the muscle is restricted. So you can see what this looks like here on a coronary angiogram, which is a special type of X-ray used with dye to examine blood flow. And you can see the narrowing of the coronary artery where this arrow here is pointing. But if we get back to our life course of heart disease, there might be a person who has some cardiovascular risk factors like high blood pressure or diabetes, but they've never had a major heart event. And preventing these is called primary prevention. And there's been a huge amount of research into this in New Zealand over the past 20 years. Improving the accuracy of the prediction. And then importantly, this has already been implemented into primary care, such as GP practices. And then one of the most common major heart events is a heart attack. And there are around 15,000 hospital admissions for heart attacks every year in New Zealand. After a first heart attack, you're at much higher risk of subsequent events. And our previous research has shown that 1 in 3 people in New Zealand died or were readmitted to hospital for heart-related problems within the first year after their first ever heart attack. So preventing these events is called secondary prevention, and that's where my current research is focused. So we want to reduce the number of these events, and we also want to reduce the inequities, particularly for Māori and for Pacific peoples, and also for women. So to address this, we're undertaking a large study of people admitted to hospital with their first ever heart attack. And this is called MENSACS, which stands for the Multi-Ethnic New Zealand Study of Acute Coronary Syndromes. And this is what my AMRF postdoctoral fellowship is based around. So we're running this study at 9 hospitals around New Zealand, and we have over 2,000 people enrolled to date. So while someone is in hospital, we collect a lot of information about their medical history, their nutrition over the past 12 months and their physical activity, their family history of cardiovascular disease, and we also take a blood sample which we process and store in our biobank here at the university. So with this blood sample, we're measuring a number of biomarkers that can help to predict future risk, including several that are in the research stage. And this is including genomics or genetics, which is my main area of interest. So I'm going to talk a little bit more about that now. So I thought I'd start with a quick refresher. Every one of us has nearly 3 billion bases or letters in our DNA sequence. We share 96% of this in common with a chimpanzee, although before we start feeling too superior to other life forms, it's good to know that we also share 60% of our DNA with a banana. So humans' DNA sequences are 99.9% identical with each other, and it's the 0.1% differences that we're most interested in. So the genetic component of your risk of coronary artery disease is estimated to be around 40 to 50%, which is actually quite high compared to many other diseases. So there's some rare single mutations that can put people at very high risk, and these affect maybe around 1 in 300 people. But for most of us, the risk comes from a combination of up to millions of different sites in our DNA, each of which has a very small effect. So what we can do is to measure these sites and create what's called a genetic risk score, and use this to predict the risk of disease for any individual. So in our study MENSACS, we'll be using these scores to see how well they predict events after people's first heart attack. So which of these 3 people is the one who's most likely to die or be readmitted to hospital? And then that person could be targeted for interventions and for closer monitoring. But it's important to remember, of course, that our genes definitely don't determine our fate. So even people at high genetic risk who have healthy lifestyles can reduce their risk by 50%. And these are also the people who get the most benefit from taking statin therapy. So another area that we're looking at is giving people different medications based on their genetic profile, and this is called pharmacogenomics. So people metabolize drugs in different ways and at different speeds, and some of this is based on genetic variants that you might have, and this is relevant for some common cardiovascular medications like clopidogrel, which you may be put on after a heart attack, or warfarin, which is an anticoagulant. So in MENS-X, we'll be looking at whether people were given the most effective drug for them after their heart attack, and also looking at what difference it might have made to their outcome if we had been able to do these tests while someone was in hospital, and then they were sent home with more personalised treatments. So it's really, really important that we're doing this research here in New Zealand, as the vast majority of genetic research has been done in people of European ancestry. So what we don't know is how well all of these risk scores and tests will work in people that have different genetic backgrounds, such as Māori and Pacific peoples. Because this is so important, we're now focusing on doubling the number of Māori participants we have in MENS-X. And I'd like to thank here the Kelleher Charitable Trust, who have contributed towards this part of the study through their relationship with the AMRF as well. Another current focus for me is heart disease in women. It's the #1 killer of women globally, which many people don't know. And it kills twice as many women in New Zealand than any other single cause. But despite this, it's still often thought to be a male problem. This is a new area of research for me, but having had heart disease in my female family members and also being the mother of 2 girls myself, I've actually been quite shocked at the lack of representation of women in cardiovascular research studies and clinical trials, and also the lack of research into sex differences in this setting. So there's a number of sex-specific risk factors in women for cardiovascular disease, and many of these haven't been very well studied because of the lack of representation in a lot of previous research. So this includes things like premature menopause, which is a risk factor for coronary artery disease. It also includes a number of things that can happen during pregnancy, including gestational diabetes, which is relatively common, but also includes blood pressure problems during pregnancy, including preeclampsia. Women are also more likely to have autoimmune diseases, which increase the risk of cardiovascular disease. So in this diagram, we have these sex-specific risk factors on the left. In the middle, we have the well-established risk factors, which are common to men and women. But also on the right-hand side, we have some what they've called here under-recognised risk factors, which kind of more of the social side relating to the role of women in the family, which is often sort of that nurturing matriarch role. And this can often lead to women putting other people's needs before themselves. And so in that case, not following up on any early symptoms that they may have of disease. Women also present with disease differently as well, which can mean delayed diagnosis and treatment. And that can sometimes be women not recognising their symptoms as heart disease and not seeking help early enough. So an example of this is a recent headline from earlier in the year about a 35-year-old Auckland woman who didn't recognise her symptoms as a heart attack. She felt silly seeking help, so she didn't go to A&E, although thankfully she did eventually visit her GP. So when we looked at how well women were represented in MENS-X, our study, it turns out we weren't doing so well, with 21% in our first 2,000 participants. And amongst these women, there were higher levels of socioeconomic deprivation than men. Women had high levels of diabetes and smoking. There was less in-hospital investigation of these women, and they had less interventions, and there were also higher numbers of women dying or being readmitted to hospital. So to try and restore some balance back into our study, we've been recently awarded an AMRF project grant, which is really exciting. So this is so we can create a new sub-study, which is called FAIR ACS. In this, we're going to double the number of women enrolled into the wider MENSAC study. We're going to be collecting data on some female-specific risk factors and look more closely at the subtypes of disease that are more common in women as well. So what's next for this research? We're continuing enrolment of Māori and Pacific peoples into MENSACs. It's been an extremely challenging couple of years attempting to undertake research in the hospital setting with COVID and lockdowns, but we're getting going again now. For the FAIR ACS sub-study, we've started enrolling women, and we've recently secured some other external funding to measure a number of hormones in the stored blood samples that we have and see that, how they relate to the age of menopause and cardiovascular risk. We've got a number of collaborations and sub-studies that are ongoing. And the nature of this type of study is that it's extremely resource intensive at the point of patient enrollment, but the value of the data grows with time. So even though we're so excited to have lots of data coming through and starting to get some results, we also have to be patient. We also have another large study, which is a randomized controlled trial looking at whether clinical management of people after a heart attack can be improved by ongoing measurement of some key cardiac biomarkers and then titrating the medications appropriately according to the results. But that's probably a conversation for another day. I'd just like to finish by saying a huge, huge thank you to the AMRF and all of their generous donors, and also to all the other funding bodies that make our research possible. All of our studies and most of our salaries, including mine, come from competitive grants, so it wouldn't happen without the support. I'd also most importantly like to thank our study participants who are kind enough to take part in this research to help out future generations, even when they're going through such a big and stressful event in their lives. So thank you very much.